Hello, everybody from Biology A. This is Miss Peachy. I'm going to be doing a lesson overview for Unit 6, Lesson 3, first semester of biology. A lot of this lesson is kind of a review from stuff we've done earlier, so it's going to seem kind of familiar to you here. So as you probably remember, as I keep reminding you, I have worksheets available for every single one of these lessons, and this worksheet is found on my website. It's pretty much just going to give you kind of an outline to follow as we go through these video recordings. And just kind of remember too, ladies and gentlemen, that these video recordings are just kind of giving you some further explanation, some overview to the unit. And you still need to make sure you read through the whole unit and do any kind of supplemental things. Sometimes they'll have little exercises for you to do, some practice, some videos to watch, and that kind of stuff. So that stuff I'm not going to go through with you today. All right, so this one is titled How Cells Make Proteins. And the first page here talks, just kind of gives you some vocab and their definitions, so we won't go through that. Page 2 is going to talk to you a little bit about what hemophilia is. So they're using hemophilia as an example of a genetic condition here that is determined by your genes. That's what genetic condition actually is. This particular type of genetic condition is called an X-linked or sex-linked trait, meaning that it is determined based on your X chromosome. So you have 23 pairs of chromosomes, 22 of them are, regular chromosomes, and your 23rd pair are your sex chromosomes, and they determine whether you have male or female characteristics. So this particular gene is found on the X chromosome. Because it is found on the X chromosome, it is more commonly expressed in males than in females. And that's because this particular trait, hemophilia, is a recessive trait. So that means you have to have a copy of the recessive allele. And if you have a copy of the dominant allele, that's going to overpower that recessive allele, and you will not show that trait. So females have XX, that's 2 X chromosomes, right? So if you have a recessive allele on one of your X chromosomes and a dominant allele on your second X chromosomes, you can still be a carrier of this trait, but you will not have hemophilia. Males only have one X chromosomes. They are XY. Because they only have one X chromosome, even if they only carry one copy, they can only carry one copy of the recessive allele, they'll always have the condition if they have a single copy of that recessive allele. So we see it more commonly in males than in females, okay? So this first page kind of goes through kind of a reminder of how everything is organized within your genes and genome, and that your genes are found in your DNA. They're part of a sequence of your DNA. That DNA is housed inside your cell. It's wrapped all up along these histone proteins into structures called chromosomes. And if you were to unwind that chromosome, you would see a double-stranded helix called DNA. And DNA is made up of a sugar phosphate backbone, and that's 2 strands that are connected to each other by these nitrogenous bases. And in DNA, that is cytosine, guanine, adenine, and thymine. Going on further, we have a process called protein synthesis. And this occurs when you take that DNA and it is read and it is transcribed first into messenger RNA and then translated into amino acids, which are linked together to make proteins. And protein is kind of what is the end result of protein synthesis. In this particular example with hemophilia, hemophilia is a condition where you lack the ability to make certain types of proteins that help with clotting of blood. So if you don't have the ability to make these proteins, your blood won't clot very well when you get injured, and therefore you will keep bleeding and bleeding and bleeding instead of having a blood clot form, a scab form, and that bleeding stop, right? And so it becomes a very dangerous and sometimes life-threatening condition if you are injured. So a protein is a group of amino acids that are linked together, and then eventually those amino acids, that chain of amino acids, is folded up into a specific shape, which is what a protein actually is. Hemophilia, you lack the ability to produce a specific protein, so you're not able to produce these proteins. Protein synthesis is how we produce those proteins to begin with. So first, in protein synthesis, the DNA is unwound and separated into two strands. Because remember, DNA is double-stranded, so we have to separate it into two strands. And that allows the cell access to those nitrogenous bases, the cytosine, guanine, adenine, and thymine. By the way, we have two types of nucleic acids. We have DNA and RNA. DNA is known as deoxyribonucleic acid, and RNA is ribonucleic acid. And they're very similar, but they differ in a couple of key ways. DNA is double-stranded, first of all, where RNA is single-stranded. DNA has a sugar phosphate backbone made up of deoxyribose sugar. RNA has a sugar phosphate backbone made up of ribose sugar, and they're very chemically very much alike. The DNA deoxyribose sugar is lacking an oxygen group, which is why it is called deoxyribose. The other difference is what forms the rungs of the ladder. So the rungs of the ladder in DNA are made up of cytosine, guanine, adenine, and thymine. In RNA, the rungs of the ladder are made up of cytosine, guanine, adenine, and uracil. So just one different nitrogenous base, which, as you can see here, are actually chemically very, very similar to each other. Now, when you're looking at DNA, remember there's two strands. So on one strand, the C is going to bond with the G. So cytosine always bonds with guanine. Thymine always bonds with adenine. So that's how DNA kind of bonds with each other. In RNA, cytosine is going to bond with guanine, and adenine and uracil will bond together. Okay, so there's a little difference because we're replacing that uracil, if you remember. So protein synthesis is made up of two parts, transcription and translation. Transcription happens inside the nucleus, and translation happens in the cytoplasm on a ribosome outside of the nucleus. Transcription is where that double-stranded DNA is opened up, so you're exposing one side, and then the DNA is transcribed into messenger RNA. So you are creating, the cell is creating a strand of messenger RNA that is the complement to the DNA. So again, that messenger RNA strand will have the ribonucleic sugars. It will have these 4 bases instead of those 4 bases, right? The next step is translation. So here, that messenger RNA will kind of come out of the nucleus through little tiny holes in the nucleus called nuclear pores, and it will find a ribosome to attach to. On that ribosome, it will be read and translated into amino acids. So here you can see the strand of messenger RNA, and you can see a bunch of other stuff. This light purple color is the ribosome, and the messenger RNA is kind of fed through the ribosome, kind of like a piece of, I always think of it like fabric being fed through a sewing machine or something, just a little at a time. It is actually read 3 bases at a time. So every 3 bases, that is read, and then it moves a little further, and the next 3 are read, and then it moves a little further, and the next 3 are read. Each 3 bases is what we call a codon. So a codon is a sequence of 3 bases, and each codon corresponds to a single amino acid. As you can see, this little structure here is called tRNA, also known as transfer RNA. Each molecule of transfer RNA has a complementary sequence to the codon. So if we can isolate this, let me see if I can zoom in here and kind of see what I'm talking about. This sequence, GCU, is a codon of messenger RNA. The complement to that is CGA. Remember, C matches G, G matches C, A matches U. So this transfer RNA right here is going to have a complement, a complementary codon. We call it an anticodon to the messenger RNA codon. It also carries on its tail one amino acid. So once it finds its matching set on the messenger RNA, it will then deposit that amino acid along that series of, in that position in the chain, forming this whole chain of amino acids, which we call a polypeptide chain. So that whole chain of amino acids, each one is deposited separately by one of those transfer RNA molecules. And again, this is also a review from a previous lesson. So every codon of messenger RNA is corresponding to an amino acid. We can use this tool here, which is just a chart showing us, it really kind of looks weird, but it helps us to figure out which amino acids each codon is coding for. You can use this one, or in the sheet that I've given you, I've also given you a second one to use, which I actually prefer this one right here. This is a circle version, and this is the chart version. So if I go back to, let's see, nope, that's not the right slide. I always get that mixed up. The left, again, mixed up. To my example from previously, we used this one, remember, GCU. So you get the codon from the messenger RNA. That's what we want. So GCU, okay. So if I go to the chart that they give you in the lesson right here, I start with this side, the left side, and I find G. And so that tells me that it's somewhere in this row, the bottom row. And then I start at the top, and I find C. And I intersect the C and the G. So it's going to tell me that my amino acid is found somewhere in this box. And then I use the right side. Remember, it's GCU, so I want the top one. And basically, in this box, every single one of these, all four of these, are coding for ALA, which is an abbreviation for alanine. So they're all coding for alanine. That's how you use that kind of boxy chart. The circle chart, my favorite, right? You're going to do it like this. You start in the middle of the circle and you find G. And that limits us to this upper left quadrant. And then you go out one ring of the circle and you find C. Now that's limiting us to this particular wedge right here. And then we go out one more to U, and this tells us it's alanine. And again, every single one of these GCU, GCC, GCA, and GCG, they all code for alanine, so it's really not a big deal here. But you'll notice some other ones are split, like U, G is tryptophan, but UGU is cysteine. It's really hard to read there, but that's cysteine. So, you have to make sure you follow all three rings. But I prefer the circle one. I think it's a little easier to use. So you can use whichever you prefer, but the circle one is there. And honestly, if this is too blurry for you, if you just Google circle codon chart, you'll get a bunch of pictures that might be a little bit easier for you to use. All right, so back to our lesson. Okay, so then in the lesson, you kind of have to go forward a bunch of slides because they have a bunch of questions for you to answer and some practice for you to do, a video for you to watch, some of the claim evidence reasoning to talk about, more practice, and then we get into this slide. And it talks about changes in DNA that are caused by mutations, and one of these affects a protein that helps to clot your blood, which results in a condition known as hemophilia. And then you can use a tool like this, which is a type of pedigree that enables us to be able to track the inheritance of this trait from generation to generation. So if you look at the key here, it will say capital X are those that have the unaffected X chromosome. So their X chromosome is dominant. They do not have the trait that causes hemophilia, right? Let's just make this a little bit bigger. If they have the Y, they are male. So there is no gene carried on the X or the Y chromosome for this trait. And if they have the lowercase x, they have the affected recessive allele for hemophilia. So we start off with Queen Victoria. She's where this all begins. Queen Victoria is a carrier of hemophilia. She has two X alleles, but she has a dominant and a recessive. So she will not display the trait of hemophilia, but she still has the gene for it. She's just silently carrying it. She marries Albert of Saxe-Coburg-Gotha, and he is unaffected. He doesn't have anything there, right? They have lots of kids. Of their children, Alice, a female, is a carrier. She's got big X, little x, so she's a carrier of hemophilia, but she does not have it. Leopold, a male, he inherited that X chromosome from his mother, so he has hemophilia. And Beatrice, a female, she also is a carrier. So Leopold's the only one that is affected with hemophilia, but Beatrice and Alice are both carriers. So Leopold passes on that little X chromosome to his daughter, Alice, who inherits her big X chromosome from her mother, Helene of Waldeck. She has three children, a female and two males. The two boys both get her small x allele for hemophilia. Both of them are hemophiliacs. So you can kind of follow this from generation to generation and kind of see how this trait is passed on using this type of tool. And then finally, we get to controlling gene expression, which is the last section. We actually did have a whole lesson on types of ways in which gene expression is controlled earlier as well. So this is just a reminder that one way in which gene expression is controlled is through something called a transcription factor. And transcription factors are enzymes, and they can be both positive and negative. Positive transcription factors will turn genes on, and negative transcription factors turn genes off. So transcription, remember, is where the DNA is actually changed into messenger RNA, right? So if that process never actually happens, then none of that protein is actually translated later at all. So in this situation, you'll have an enzyme that will actually stop transcription from happening or cause transcription to happen. And that's important to regulate transcription, because even though you have a complete copy of your DNA in every cell in your body, you do not want every cell in your body to make all the proteins that your DNA is coded for. That would just wreak havoc everywhere, because certain parts of your body do some functions and other parts of your body do other functions, and so we have to limit what is being transcribed in the regions where it's actually being used. All right, so that is in a nutshell, ladies and gentlemen, what we learned about in lesson 3. If you have any questions on any of that, you can feel free to email me and set up an appointment to meet in my live lesson room, and we can kind of go over some more of it together. Have a great day. And get my mouse to work, but we'll talk to you later.